Forward to the last presentation of this session. Scott McNeil from the National Characterization Laboratory in US, and he has a very short title, the US NCL View.
Okay, thanks. Well, in trying to select what I'm going to talk about with respect to the NCL's view in respect to ADME talks, I came up with something that's been talked about now several times, and that's critical attributes and how to get there. It's always dangerous just to give NCL the floor for any topic whatsoever. So as we just discussed just now, I think it's pretty well established that physical-chemical parameters will influence biocompatibility. And we've seen over and over again in my laboratory as well as the literature that even changing things like zeta potential, the length of peg, the size, you get quite different changes in pharmacokinetics. You can take advantage of those, and ideally if you see an adverse effect, you can engineer around that effect. So, without exaggeration, we can almost dial in the properties that we want on the right-hand side. Here's an example that we've used at a case study in the past, if ever you've seen our lessons learned, but it really drives home the point that I'll make today, in that with different batches of material, there can be just slight differences that are really hard to pick up, but they can have very, very dramatic effects in vivo. So here's an example where we had two batches of material from a sponsor, and they were fairly far along as far as their development. But we saw dramatic differences between batch 1 and batch 2 in that batch 1 caused severe lesions and, in fact, would kill animals, lesions in the lung. But whereas batch 2 revealed no toxicity. It was the same company that was producing them. They had the same processes in place. So why the differences? And could we then do a retrospective study to look at what PCC difference there was that could point to a mechanism of action. So if any of you have followed NCL, you've seen this example many, many times. But what we were able to do is it pointed to a difference in the surface chemistry, and specifically we thought that the peg layer may have something to do with it. But we found out that through our traditional method, we did DLS and other things, we were not able to pick up a difference. But you truly have to know the measurand that those instruments are recording. And in the case of DLS, it looked at the hydrodynamic radius, where the two species looked identical. But then when we went into further analysis with the density of the peg layer, as you can see up here between batch 1 and batch 2, I trust that you'll be able to read that gel to see that for batch 1, most of the peg was in the supernatant as opposed to still attached to the particle. That's contributed to the mechanism of action in the toxicity, in that the peg was disassociating from the particle. Why have I taken so long to explain this? It's because this is germane to critical parameters, and it's unique to this particle. Those critical parameters are things that if you change even slightly, they have a pronounced and amplification effect during the in vivo response, contributing to either safety or efficacy. And we're often asked, and the FDA is asked, well, what are my critical parameters? Well, you're the one that's supposed to tell them, as I said yesterday. Okay, so for that formula, it was surface ligand density. There's various methods to begin to look at your critical attributes. If you think of it, if you're engineers, you can think of it as look at the total derivative and what partial derivative and if you do a sensitivity analysis, are most important in contributing to that total derivative, the total derivative being safety and efficacy. So there's various ways to quantitate the nanoparticle coating, as I'm showing you up here. I just gave you an example for monitoring PEG. We can look at batch-to-batch heterogeneity and where the drug is within various species in a batch. We can do an immunosafety assessment, and we're having some great success looking at in vitro and in vivo responses there. And again, looking at how PCC changes immunotox. And fractionation, looking at where the active species is. Is it bound? Is it incorporated into the plasma? Is it released immediately? And that can be very important as far as, well, both safety and toxicity. So for surface characterization, there's the reason why you want to characterize the surface. The regulatory agencies are going to ask you for it. They're going to assume that you have very good understanding of that surface chemistry and the boundaries for lot release criteria. So for example, in the previous example, if you knew that anything less than 60% pegylation would cause that aggregation in a lung lesion, how close to 60% can you get? And what are those boundaries for lot release? Or do you have to keep your lot within 80 to 100% and so on? That's the type of things that you have to define as you go into the developmental process. And there's quite a number of methods available for surface coating if you can strip the surface coating off. If you have a gold particle, it's very easy. If you have something like a liposome, then it becomes to be more complicated. There's methods available to you. Any method that we have is on our website. Feel free to access it and use it as you see fit and modify it as long as you can validate it. Here's a few techniques for coating quantification, ones that we're now getting into more and more. Ideally, it is a technique that we developed that they can be ported to a CMO or a CRO. We can come after some of these problems with some very exquisite toys. We have a very nice tool set at NCL and available to us through Frederick National Lab. But our goal is to make something that's available that a CMO or CRO can use so that they don't have a cryos electron microscope. There's no way that they could afford, a $6 million instrument for process control. So you can use things like CAD, charged aerosol detection. We use these in line with other LC methods. TGA is we're using more and more. One thing about TGA is you look at you're essentially burning the material and looking at the weight loss. You do need a little bit more material for a TGA than you would for other proof of concepts, but presumably you're at that level once you're in GMP production. And then various classical chemical methods, Elman's reagent and doing thiol reduction, and as I showed you earlier, stripping off the pegylation layer in that one specific case. So also attempt to use orthogonal technologies where you're not chasing artifacts, that you know that the measurand that you're monitoring is confirmed by another technique. What about heterogeneity? This is really quite an interesting challenge to us that we've had over the years. It took us many years to be able to address this. So the problem is if I see multiple peaks on DLS or any other method, and I have a therapeutic, where's my drug? Which species is my drug in? Because each of those species will have different distribution profiles. And so am I optimizing an artifact? Or am I optimizing and am I truly tracking the species that goes where I want it to go? So a question I posed to my team over 5 years ago is, find me an answer for that. And they said, there's no way we can do that. Well, we finally were able to do that. Jeff Clogston in the lab through FFF was able to track which species that you see on something like a DLS actually has the drug loaded in it. And that was a question that FDA is now hot on and asking, and I assume EMA and other regulatory agencies as well. Under immunosafety, if you're in the cancer field, it's actually not required by the agency until you're in phase 2. That said, identify these types of things very early. Some things that we've had trouble with now that we've characterized over 360 species, what you don't want to have happen is you get a call in the morning and say, hey, 5 out of 6 of your mice died in this specific arm, and we don't know why. They died within about 4 or 5 minutes. And so then you begin to rule things out. You don't want to just repeat the study because you're just killing mice. So we've had things such as thermogenicity, or DIC, disseminated intravascular coagulation. We've had hypersensitivity reactions. I mentioned yesterday some of the problems we've had with endotoxin contamination. And there's also an exaggeration of an effect due to nanoparticles, especially if you have contamination. This is actually very important, is there may be a threshold that FDA requires as far as what's allowable for endotoxin contamination. But what we've seen is that a nanoparticle gives you a greater than additive response because it uses another pathway for internalization that causes an amplification of that cytokine storm. So even though you're within tolerance, you can see things like a cytokine storm where the animals will die on you. They're due to hypersensitivity. Here's Marina's written several papers and books on this. There's a lot of material on this. If you're interested, please look it up. We have most of our bibliography online at the NCL website. Under nanomedicine fractionation, and this is very relevant for comparison of follow-ons and biosimilars and nanosimilars, is what is the actual species that you're comparing in order to do an apples-to-apples comparison. And this method actually begins to get at that, is oftentimes we'll monitor the total amount given, the total dose, and assume that as long as you're monitoring the total dose between two species, you can make assumptions and comparisons. Well, that's actually not the case. In that what happens is they're in equilibrium, that a legacy version of a drug is not the same as something like a liposome that has a different release kinetic. And so the true species that you want to monitor is this right here, the free unbound. That is the species, for most cases, that is available, that is the toxicant as well as the efficacious species. So we now have a series of assays to go after this. I think I have it on my next slide, but keep this in mind, especially if you're in the generic field, is what are you comparing? You're not comparing the total dose, you're comparing the free unbound that is available to do its action. Here's a few assays that we've looked at over the years. Some have their strengths and limitations for each, as far as cost and what species they look at. Please feel free to grab that control from Journal of Controlled Release. Okay, so in summary, hopefully you know what critical attributes are now, and they're unique to your particle. They can be almost anything as far as PCC and process control, and they can be identified by using some of the tests that I've shown you up here, and also they certainly help out with batch release criteria as you scale up. With that, here's the real smart people in the lab, and I'll close and thank you for your time.
So thank you. First, I would like to thank you, Scott, for sharing this data, because I think you see so many different nanoparticles, and it's so important that people really see examples of heterogeneity and the problem and challenge of reproducible nanoparticles. Questions?
What's the status, Scott, of mapping the release rate from nanoparticle that partitions to protein, but opposed to being free, and sort of the assays for release overall in nanoparticles?
That's a great question. So we have a number of them. I wasn't able to go into depth in there. But what we see is that you cannot assume that they're equivalent, even between two liposomes, for example. So there's assays available where we put it into blood, for example. If it's a lipophilic molecule, that's probably one of the best ones. Otherwise, it's very difficult to track it. It'll often just pop out unless you tweak it. If it's in a liposome, a hydrophobic molecule. Other molecules that we can use will often dual radiolabel. So we can look at if the drug comes apart from the nanoparticle, and we see that often. And other methods are just doing a solid support method if the particle will survive it. And again, it depends on which measurand you're going after. As long as you don't care if the particle falls apart, you can use that. Steve Stern in the lab has published a number of these, and his article in Journal of Controlled Release is a great starting place.
One more question.
Thanks, Scott, for this nice data you presented. Just a question. If you look a little bit more into probably eventually clinical application of such drugs, what would you suggest if you need two different drugs with two different nanoparticle design? Do we have any idea what they are doing together in a body?
So you're talking about coordinated PK. The greatest example of that is the Celator presentation, where he does coordinated PK. He's able to match and exceed the therapeutic index, or at least decrease the number of deaths by doing that. There's quite a bit of background experiments that you have to do because the physician is going to like to be able to modulate both of those. So you have to make the case that your fixed ratio is the best. And then that, again, coordinated PK eventually is going to have some desired effect, either decrease toxicity or increase efficacy. It can be done, and the successful story is there where, again, as they were just bought out for, what was it, $1.2 billion. Yes, so there's a lot of room to maneuver there.
Okay, I think we stop. I thank all speakers for the presentation and also for keeping on time, and the audience for having a lot of interesting questions. Thank you.